Hi, everyone. Welcome to Foresight's Health Extension Group, sponsored by 100 Plus Capital. I'm really excited today to have Owen Phillips here from brainkey. And as the name says, he'll be talking about brain aging as the key to longevity. I'm going to share a lot more info about you in the chat, including links to your work and so forth. But in the interest of time, and since I expect there to be plenty of questions about this topic, I'm just going to give the stage up to you. Thanks a lot for coming. Really appreciate it. And I'll be in the chat monitoring for questions. And then we have a Q&A. Thanks a lot.
Yeah, thank you so much for having me. And I love casual talks and Q&A more than I like just speaking. So please throw out questions or even just start yelling at me. That'll make it more interesting. Why I say the brain is key to aging. So what I see in the longevity field is everybody wants to live 100 years plus, 120 years. But what I keep going back to is, what do people die from 80 plus? And we'll talk about that more and more. But what gets me so interested about the longevity space is the brain is so vulnerable to aging that whatever you're doing in the longevity space could potentially have an impact on the brain. So this is where I'm going to go with this talk, but I'm going to break down exactly why it's having that effect, and we'll do this in a more organized fashion. But in the meantime, let me just give you a quick background on who I am. And yeah, so what is the leading cause of death for people over 80? And we're just going to ignore COVID for a little bit and the potential neurological effects that's having, but generally, brain disease is the top killer for people over 80. So if you do want to live to 100 plus, you should really be concerned about brain health. So where I'm coming from with this is I'm a neuroscientist with over 50 publications. We have a team at brainkey that continues to publish frequently. We just had a Nature publication by our CSO. We love just staying up to date with the latest neuroscience research. So everything we do is informed by what is happening right now. And that goes into how we think about brain science and how we build out brainkey. So from a high level, we're going to talk about why the brain is very vulnerable to aging. The biggest thing for this is the brain is so energetically expensive. So everything keeps coming back to why the brain is energetically expensive. And we'll talk about the reasons why, the compute, the wiring, and then we'll talk about how the brain gets all its energy. What does it do when you burn all this energy? And then just how do you protect the system? And then we're going to talk about how do you measure these overall large systems and then what you can do about it. So that's way too much to cover in 30 minutes, but it's very high level. And then we can maybe get into a little deeper in the Q&A. But I think there's something that always comes through with the brain is that people bring so much to it. Either they are deep in the discipline and they're very narrow focused, and then you get so into the weeds, you don't see the whole picture of what's happening in the brain. That's okay. That's incredibly valuable and interesting. But if you're not part of this field, you might come to thinking about the brain as kind of the most common thing we come across is that people think of it in these kind of older school way of thinking where very specific regions have very specific functions. It's very neuronal based. And that's not the way I think about the brain, and more people think about the brain. It's more of in a systems perspective. So these entire large systems, how they interact, how that affects how the brain functions. So that's what we're going to focus on here is these large systems, try and take a step back and then view the brain from this outside perspective. So the key thing here is that the brain is very vulnerable to aging. Okay, so I was trying to visualize this in the best way that I could, and my wife thought these were silly, but I wanted to share them with you anyway, which is I've got a brain here, I've got a body here, and I've got a brain in the skull, so that's where it belongs. But what I want to show you, just to try to impact visually, is that the brain is very small by weight. It's really not too much compared to the rest of the body. It's only 2%, but this is proportional in this image. Again, it's a silly image, but the brain is burning between 20 and 25% of the brain's energy. So that's just a tremendous amount of energy for this very small structure. So what happens? So the reason I think this is so important is you've got this structure just burning a tremendous amount of energy. So to me, the entire body is just really built around providing energy and support for this brain that you have. And Everything else is more of a support structure for your brain. This is key because the brain isn't storing energy, so it needs to take in all of the energy it's getting from outside the external body that's shuffling it into the brain. So again, the metaphor here is it's a lot like Hawaii or an island where you need to get in most of the resources for it to function. So without that, if there's something going wrong outside of this brain system, it's going to affect the brain very acutely. That's the key point I want to, from a high level, get across is the brain is very energetically expensive. And because of this, it's going to be very vulnerable to aging. And again, it's a number one killer for people 80 plus. So if you can make it past all those acute causes of death, like heart attack, stroke, or cancer, then you get 80 plus and your brain aging really kicks into gear. And of course, for younger people, it is a problem as well. My mom has dealt with issues and she's not yet 80. So it's definitely an issue for people below 80. But I just want to emphasize that it's number one for people 80 and above. Okay, so if you're burning tremendous amount of energy, that's going to put a lot of strain on the system, both with waste products and even heat, heat is the thermodynamics of the brain is not getting enough attention, gets more attention in Japan, but that's also a very interesting thing that we'll touch on very quickly. So what do we got here? We've got billions and billions of individual neurons, and I think everybody's familiar with neurons. But one of the interesting things about them is that even at rest, they burn a tremendous amount of energy. So you can't just shut off your brain. So it's continuously burning energy. And you have to always supply that. If you mess up that supply system, then that's going to cause downstream effects. The other interesting thing there is that if you are using your brain tremendously, then you do start burning many more calories. The common anecdote here is that chess players can burn as many calories as maybe a marathon runner when they're in tournament level competition. So you can vary how much energy you are using with your brain, but generally it's around 20 to 25% of your energy expenditure. And neurons are what is so heavy there. What is, I think, missing in this equation, and one piece that I really think is important for aging, is the connections between the neurons are primarily done with oligodendrocyte cells that myelinate the axons. But one thing that's interesting here is that once they're built, they're relatively cheap to maintain the fat around the axons. So I'm just skipping through tons of neuroscience here, but the way I'm thinking about this, so you've got these computers and then you have the wires connecting different parts of the brain. So right now we're talking about the wiring in the brain. And once that wiring is set, it's relatively cheap from an energetic perspective. But what it's not cheap is to build, and it's not cheap to repair if there's damage. So keep that in mind with aging. So with aging, if you do have energy deficits and you have trouble with your wiring, that's going to be very hard to repair. So I think that's something that gets missed in terms of neurological disease is even if you want to develop a therapeutic that targets neurons, if you don't address this problem that you have with the brain's wiring, for older populations, it's going to be very tough to remyelinate. So that'll be issues there. Okay, this is my fun graphic again, but this is for the developing brain. Anything we're talking about from an energy perspective is just amplified in a developing brain. I know we're in a longevity conference or a longevity talk, and we're talking about aging as long as possible, but if you have a developing brain that is not getting the correct level of energy supply, say you're pre-diabetic or you're just not on the right diet, it's not for every patient, but just in general, that's probably going to impact the brain development. So just think about what I just talked about, the myelination of different regions. That's very energy heavy. If you don't have the right energy delivery system to your brain, then you're just going to impact that system at a very critical point. So if you're impacting how your brain develops early on, that's going to impact your longevity in the long space. Just from a societal standpoint too, I think that's a very interesting conversation that should be had more and more often. It's about just how we are providing nutrition for a developing brain. I have a few publications on this that don't get a lot of attention, but I think it's a very important topic. Okay, so just blasting through everything. So feel free to ask me questions. I can't, but there is chat. Okay. If somebody has a specific question, let me know. Okay, yeah, there's nice chat going on. I can't see the specific question without stopping everything. But okay, the vasculature, again, I've been doing neuroscience for a few years now. I originally came out of UCLA and laboratory neuroimaging there. But when I first started, there was very little attention to two things. One was the brain's wiring, which I was just showing you. And then two is the vasculature of the brain. And what's really exciting is now these are getting so much attention, especially the brain's vasculature, which plays many roles. One of the obvious ones is it's shuttling glucose, so the main supply for the brain's neurons. But the other thing that I think is not quite addressed enough is the timing. So how quickly and just where blood is delivered is very important. So if you're doing some sort of task, say playing chess again, and you need to use particular parts of your brain or maybe recall a move you saw in the past, you're shifting where you're using parts of your brain. And when you do that, you're shifting where blood is going so that those regions can get energy. So that's a really fine-tuned system. So if you do have problems with the underlying vasculature, just the physical structure there, that'll impact how blood is delivered. So one thing that's interesting that we're talking about is this has this cascade effect where actually the brain's wiring is very vulnerable to energy disruption. So if you're not getting the correct energy into a system, the brain's oligodendrocytes will suffer. They can actually suffer and then cause downstream effects on the neurons. So all these systems are very tied together. Okay, this is another system. Again, when I started neuroscience, almost all the focus was on neurons. Very little was on the surrounding systems for the brain, but this system is getting more and more attention. The brain's, from an easy way of talking about it, is the brain's waste removal system. So if you are burning tremendous amount of calories just all the time inside your brain, you need to get rid of waste products. You need to get rid of the heat that's being generated from that. And this is where the brain's fluid system kicks in and needs to essentially funnel that out of the brain. What I'm showing here is a patient with, in this, actually we can see, okay, that white area in the middle of the brain here, these are ventricles on a T2 brain imaging slice. Yeah, I hope people can see that. I'm pulling my mouse over it. And then on the patient on the right, we have a late stage NPH patient, normal pressure hydrocephalus, where this patient, that waste removal system is not functioning correctly. It's building up in the brain and actually causing structural damage. I wanted to show this to emphasize this is a very extreme case where this system is failing, but we think, and we're seeing this in more and more patients, where this system isn't functioning correctly in more patients than this very extreme patient right here on the right. This is a system that was actually functioning abnormally in my mom, and she ended up having surgery to put a shunt in her brain and end up draining that fluid out of the system. I know we're in a biotech group here. I think there's a tremendous amount of opportunity if you were developing a therapeutic to target this system. Right now, the main therapeutics for treating these type of patients is a neurosurgical procedure that's expensive. And it's pretty barbaric if you think about it. That's the best way we can treat these patients, is actually providing brain surgery and putting a tube through their brain to drain out fluid. It's pretty old fashioned to me, even though it can be very effective in patients. So my mom has responded to it very well. Okay, so we've got neurons, we've got the wiring, we've got the energy supply system, we've got the waste removal system, and of course, here, actually, let me show you something fun really quick. Okay, so I've only been showing brains for the most part. And when people think of a brain, what they're thinking of is this top part of your head, but it's not. It's not a Marie Antoinette and we guillotine the brain, and that's what we're looking at. The brain is the whole system. So I think that's something that we're starting to do at brainkey, and I think is very important to start thinking about the brain as an entire system. it doesn't just stop, at your neck. It continues downwards. This becomes very obvious with the brain's waste removal system, which I'm highlighting right here in blue. This is the fluid system that is used to take out waste products, but also keep the brain cool and provide cushioning. So you see it very clearly once you start imaging it as an entire system, that the brain isn't just, the top of your gray matter or the brain's wiring. It's the whole system put together and have an impact on how your brain's going to age and the cognition down the line. I really love that conceptual thinking about the brain rather than the brain as just this piece that ends at your neck, because that just forces you to think about the brain as the system rather than just this discrete thing that just ends at your neck again. Okay, let me see if I can find the sharing. Okay, I managed to cram a bunch into a short amount of time. Let's see what else we can talk about. Okay, I'm not going to go too deep into the immune system. It, again, interacts very much with the brain's vasculature, which does function as the blood-brain barrier. And so if that isn't tight, again, with aging, that becomes leaky, for lack of a better term, and then more things can make their way into the brain when they're not supposed to be there. Then there, of course, there's more to that than that. But what I want to touch on really quick is that, okay, we have these very large systems, but now we can combine what we know about the brain and also with the tools to measure how the brain is aging. And primarily what we do at brainkey is we use brain MRI imaging, and we're able to get very nice pictures of patients' brains. And then we can examine how the systems are changing. From a high level, what we look at is brain age as a biological age clock. We can provide a single score or a patient score, how their overall brain health looks. And in the neuroscience literature, this has been used over 10 years. There's various algorithms that basically fit population level data together and then try to map how individual brain is aging against that population data. It's a nice overall proxy. It's non-specific, but again, it can give you a nice general sense of how the overall brain is functioning or aging. But we like to take it further. So we have this overarching score, but then we want to know what's going on at these individual systems levels perspective. So I'm just going to show you quickly what we're doing at brainkey and how this allows you to actually measure how these different systems are aging. Let me pull this up. Okay. So if you haven't seen brainkey before, this is how our visualization looks for a physician or a patient. In this case, I'm looking at a physician view, but we have the brain visualized in 3D. So we're taking the brain images from the MRI scanner. We're applying our algorithms to the data. We're pulling out the brain and then reconstructing it in 3D. This provides a great visualization, but also allows us to get data on various parts of the brain. So I'm looking in this particular area that's very neuronal heavy. I'm looking at the frontal lobe, and I can see how they're changing over time. So how that patient is actually aging, how they compare to the overall population. So I have this metric now, and I have lots of different metrics about this. So I can get a picture of how this patient is aging with their neuronal health. And various structures can have different impact in terms of the neurological disease, but I prefer to think of it from an overarching perspective of how are these impacting these large systems. So things like the frontal lobe gray matter or the hippocampus can tell me things about the neuronal health of the patient. Again, so this is generally what we do at brainkey is we take in the imaging data and then we try to look at these individual systems. We've got about 3 separate reporting now. I think I've got 2 minutes. I can quickly show you what this is looking like. Oh, maybe I can't. Okay, I don't have that up right now, but so that's what we try to do is we try to take in the imaging data and then show you what's happening inside the brain and try to map that back to these individual systems from that perspective. And where this becomes useful from a clinical perspective, what we see is that traditionally you're looking at the brain from a perspective of a disease. Does this patient have frontal temporal lobar dementia? Does this patient have a vascular dementia? Or does this patient have Alzheimer's disease, the big elephant in the room? trying to pull this back and understand what is the system that's really faulty in this individual patient. Is there multiple systems that are faulty in this individual patient that are contributing to their overall brain aging? So it doesn't have to be just about differential diagnosis in the patient. I think that's a late stage thing that most people are looking for, trying to pull out. But what we're really about is understanding how the brain is aging in an individual patient, not looking for the diagnosis of that patient. If we get the diagnosis, say in my mom's case, we were able to take her data. She was getting pushed towards an Alzheimer's diagnosis, even being enrolled in a clinical trial. But we were able to take her data and look at it and come up with that she actually had normal pressure hydrocephalus, the specific variant that is, in this case, treatable. And so for people like my mom, we can provide this information to help their treatment. But we really want to understand it's not just the disease diagnosis. What was wrong with her was that her brain's lymphatic system, their brain's waste removal system was faulty. So we might put that label in a clinical perspective as normal pressure hydrocephalus, but the underlying biology that we see is that there was a problem with the brain's waste removal system. So that's where we're trying to take neurology, is from disease-specific diagnosis to more of a systems biology perspective. Okay, that's what I've got, and I don't want to keep boring everybody, but I hope this was interesting to you, and I'm happy to answer any questions or go deeper into any of this.
Okay, wonderful. Well, we'll kick off with Nicola, and then I think we have Christine already in the queue. And so just a reminder that if anyone wants to ask any question, feel free to either raise hand or drop your question in the chat. Okay, Nicola.
Thank you very much for very nice talk, Owen.
Yeah, we should have gone deeper into immune stuff.
I can ask you a deep question about the system. So you guys are having a very interesting data that is unlike what is out there in the clinic. In the clinic, things are usually made relatively quickly and they're running towards the diagnosis. But you have very detailed reconstructions of the structure of different elements of the brain, which are way better than what you can get with the clinical reconstruction and registrations. So I was wondering if you take a patient and you can follow them in several years, and you look at the relationship between their brain regions and how different subregions of the brain degrade at different paces. So everything seems to be always having a level of atrophy. Sadly, the direction is the same for all the parts of the brain. But if you can look at the patient and see if the subregions are aging at different pace, if you can make a prediction with what kind of problems they have. If, for example, their motor system is going down faster than it should be, is it possible that they're too much sedentary? If their auditory system is going down, are they going to develop auditory problems when they age? Do you have that level of resolution?
Yeah, so I'm going to answer your question, but then kind of go on a rant. I'll do the rant first. So this perspective of a particular region of your brain has, an impact on a particular function, I think we're too attached to, and I think really comes out of our early education in neuroscience that just gets hammered into our head, I think, from an early perspective. And it is valuable, but if I do think of it at all anymore, it's generally, it has to be an acute case. Obviously, if you have a TBI, we're talking about something different, but the last brain regions to develop are going to be the first regions to go, is generally the assumption that often plays out. So regions that, like your frontal lobe or executive function, develop last, that'll probably be the system that is most vulnerable to aging and start kind of going away first. And you see this in patients, very acute patients, where they might still have respiratory function and the ability to swallow, but nothing else is working in their brain, or it's pretty totally gone. Okay, so that's one quick thing. The clinical data that's collected, if you're just going to a hospital and they scan you, in the emergency room or even the scanner at most neurology clinics, you're not getting the best images acquired from your brain. And we work with high-end clinics, and they'll send patients to some places and doesn't depend on, so why I'm bringing that up, it doesn't depend on how much you're paying for the MRI scanner for how good the images are. Generally, it just varies from location to location of how much effort they put into acquiring quality images. But because we get data from all over the world, we've had to develop our algorithms to adjust to very low quality data. So that's why we can reconstruct these various brain regions, even if it's just a really crappy clinical scan, patient is even moving. So that does open up kind of what you're hinting at, Nicola, for being able to differentially diagnose these patients or track over time.
Yeah, so my thought was more into, can we integrate this into your approach with systems? Because, for example, the different lobes of the brain have blood supply that comes from different arteries.
Yeah.
And if someone has a poor blood supply in the temporal region, for example, we know that people who have smaller temporal region might end up with poorer memory, for example. So is there a way for you, looking at the structure and how that is evolving, because you have an insight inside the brain, you're looking at the details of the brain of those people. So can you relate and be like, oh, wait, you might be having a problem in your blood supply in the temporal lobe, but it's shrinking faster than it should be. So maybe let's look at your, I don't remember which artery supply.
No, I mean, 100%. So that conceptual idea of combining imaging with this other domain knowledge, which is the brain's vasculature, I think is so important. In general, we've just isolated these various domain knowledge and not combine those. And so that's what we're really pushing forward to do. So I really love that thought, and that's what we're trying to do. And you guys have some cool papers. They're right. Or you're working on some stuff. Yeah.
Okay, Nicola, are you satisfied for the moment or?
Perfect. Thank you.
Okay, cool. The next one we have Kristen.
Hi.
Hi, Kristen.
Thanks for a great talk. I'm a neuroscientist as well. And we work a lot with postmortem brain biological ages using multiomics. And so my question is more of a technical one. So do you have a graph that shows your R squareds with the gray matter volume versus chronological age? Or maybe a paper you can point me to? I'm just curious.
Yeah, I can follow up with that for sure. If you want to ping me, we work a lot with James Cole out of UCL, who's at King's College. And so he does a lot of brain imaging, brain age papers. So yeah, but we can talk further about the underlying algorithm there and the data. But the postmortem data, I think we should be working closer together. That is just a fantastic resource. Not easy to get a hold of that for obvious reasons, but very useful too.
Yeah, I would love to talk more about that. I don't know if you have an email you can share in chat or whatever, but yeah, I'm fortunate to have done my PhD in one of the biggest brain banks, so it is hard to get your hands on that.
I'm not encouraging everybody to go do this, but if you can hold a brain, yeah, again, I'm an underlying biologist, so I'm okay with this type of thing. But if you hold a brain or just interact with an actual brain tissue, it'll really impact how you think about the brain. At least it did for me, because it becomes not this philosophical thing you think about, but it becomes this physical structure that has constraints, and physics has an impact on it. It's soft and squishy, so banging around and playing sports where you get in the head, which I grew up doing, it's maybe you might want to think twice about. So actually physically interacting with something like that can change your perspective of the brain.
Absolutely. I've done a few dissections myself, so I can attest that has a big impact for sure.
I would say for all of us lay people, don't try it home.
We do 3D prints of the brain on occasion. It's very different. It's plastic, but at least you can get an idea what the brain is like.
Oh, damn. That's also a good memento mori, like bit to have on your desk. Okay, next one up we have Jason.
Thank you. Thank you a lot, Dr. Phillips. Really great presentation. I was just wondering in terms of, when you're looking at the brain, I know you're seeing a lot of biomarkers and coming up with a biological age for the brain and seeing that, patterns over time shift. But what I'm really wondering is about just purely like the size or the volume of the brain, we see this pattern that over time it just gets smaller, it shrinks. And so do you have any thoughts on, if, in terms of that biomarker, just purely the size, just shrinking and gaps forming, leading to cognitive disorders? Any thoughts on how we go about sort of massively slowing that down or reversing that pattern?
Yeah, so we haven't come across an 80-year-old or a 90-year-old that doesn't have atrophy in their brain. So that's again where everything I see, the brain is very vulnerable to aging. So how do we impact that? There's lots and lots of research where these fun things like exercise, diet can have tremendous impact on brain aging. But of course, we're generally looking for a more foolproof solution there. What I think for an individual patient basis is if you can understand that if you particularly are vulnerable with, say, again, like my mom, your waste removal system, the brain's lymphatic system, maybe you can emphasize that system and try to do something targeting that. So in my mom's case, in retrospect, when we look back at her, she had massive problems sleeping. And sleep is very critical to that system. That's when the system really, kind of takes over and does its job. And so if you're not sleeping, that system probably isn't functioning as well as it should. It's not even getting the opportunity to function as well as it should. So why I brought this up, if you do know the underlying problem for a particular patient, then maybe you can target that in particular and go after that. That's relevant for biotech companies as well, because if you are developing a therapeutic that has a specific target, you want to get the right patients enrolled in your trial. So you don't want to get just a general population of Alzheimer's patients. You want to get a population of Alzheimer's patients that have a specific phenotype or something specific is happening in the brain. And so your drug can target that and have an impact. I would say for a lot of the Alzheimer's case failures, this lack of differential phenotyping has contributed to failures within those trials. So my mom, again, is a great example of that, where she was being enrolled in an Alzheimer's trial and was passing with flying colors through, but that wasn't necessarily her underlying cause, and she wouldn't have responded to that therapeutic as effectively as maybe another type of patient.
Right. There's so much difference between everyone's brain. I mean, in terms of the ability to, say, regrow some of the brain, I mean, how would you think of, say, a highly invasive treatment versus maybe a non-invasive one to do some kind of brain regrowth or neurogenesis to sort of...
For me, right now, I wouldn't cut into the brain or do anything invasive, but it works for my mom. But I find that really middle ages, for lack of a better term, that's the best we can do. Is pretty embarrassing that we're not doing a better job treating patients. In general, this is such an issue in neurology where we're not offering enough for patients, and the drugs that are out there or getting approval are generating so much hope, but they might not have as much impact on the patients as they would want. So that's the shitty thing. The nice thing is there's so much opportunity in that space. And I think over the next 10 years, that's going to have the most impact and be very, very good for the people who go after that in a smart way.
Oh, sure. Thank you. Appreciate it.
I think, Jason, there's more we could go in there, but that's my quick take. So appreciate the question.
Oh, yeah. Thank you. I'll follow up.
Next up, we have Larry.
Yeah, hi, Owen. Yeah, good presentation. I mean, NPH, I had a good friend who had it or didn't have it, but he got diagnosed as it, and they ended up putting a shunt in him, and he probably ended up having Lewy body disease, and he's just deteriorated since then. Are there any, good early diagnostics? So, because it's either missed a lot or it's sometimes misdiagnosed, because a lot of the things they look at your gait and they, it's really a lot of physical.
Oh my God.
Yeah. Then the whole flow, what controls lymphatic flow or cerebrospinal fluid flow out of the brain, I think, how to get at that is, I think, really important. It'd be good to hear any insights you have on that. Yeah.
So I've given talks at Stanford, University of Toulouse in France, and Japan on NPH. And what's fascinating to me is everywhere you go, even from locality, from UCLA to see Stanford, how people think about NPH is wildly different. So that drives me nuts. But this is a continuous problem in neurology in general, where there's just not unified things.
Yeah, this friend of mine got treated
but-
at Hopkins, and they put the shunt in, and he didn't get any better. You know, it was really
Yeah, I mean, it's a risk. I mean, my mom responded, so I'm not going to, I mean, it's affected our entire family.
Yeah.
That was just horrible. And so
Yeah.
it's changed everybody's life. So we're so happy about that. So that's fantastic. But yeah, we could do a better job identifying these patients and identifying these patients early. And then it's misdiagnosed 2/3 of the times, but then how often is Lewy body disease missed? So it just goes back to the problem, which gets me in trouble if I go too deep into, which is just the way we diagnose or what we call particular diseases is based on, say, Alzheimer's disease was really developed the disease criteria first in 1906, and that's still what we use all the time for everything, and everything gets referred back to these things. And it's very hard to break out of that way of thinking, but I think it's starting to happen. And I think that'll open up more pathways to therapeutics. From the pure startup perspective, I'm not going to bash disease diagnosis because those are so ingrained in our healthcare system. And if you are developing a therapeutic, you might definitely go after that, or even developing a differential diagnosis tool for use in the clinic. I wouldn't disregard that, but from a biologist perspective, it drives me nuts. Cool.
great. Yeah.
More problems to solve. Okay, next one up we have Liz. Oh, Larry, did you want to follow up? Sorry.
No, that's fine. That's great. Oh, no, that's good stuff. Yeah.
Okay, Liz, you're up next.
Hey, really exciting. Actually, about 21 years ago, when my husband was doing his master's at the University of Washington, he worked on this technology. They did the first 3D imaging of a brain from multiple imaging sectors. And so he was really excited to be part of that. He was actually in the electrical engineering department, but he specialized in computer science. I mean, he works at Adobe to this day. So anyway, so excited to see companies using this. But my question is, of course, how can companies like mine effectively, efficiently, and not too expensively use your technology in both medical tourism, which are investigator-led studies, and clinical trials? Because we are awaiting the response for our first pre-IND for dementia through the FDA. And so we'll see what it says. Are you working with companies to actually move this forward?
So we primarily work with, I guess, longevity clinics. So high-end clinics providing usually a detailed profile on the patient, whole body. And so we come in as the brain module. And so we can help companies get set up to do that and then provide the analysis, the reporting, the 3D visualization, and all of that.
That's cool. I'm consulting companies, not companies, but clinics to move towards longevity. So we're bringing in longevity testing to a variety of clinics throughout the world. So this might be great.
Yeah, no, we work with a lot of clinics in that space, and it's growing very quickly, which is really exciting. And I saw the Wall Street Journal had a nice piece on longevity clinics. So it's good to see this kind of getting some awareness.
Do you have a general cost around this? Because I think that's, the most important thing is, especially investigator-led studies, keeping the costs as low as possible for patients is essential to the success.
Yeah, I mean, we could talk further about that. My family history doesn't come from high net worth individuals. And we're developing tools primarily that are used for high net worth individuals. We prefer our tools to make it to the general population. I think that's so critical from a population perspective, especially if you're talking about overall healthcare costs.
And
we have to do what we have to do as a startup. But in general.
It's hard to push innovative medicine forward if there isn't a lot of data around it. So let's reach out to each other and please consider, because, that sort of technology and that sort of brain imaging. I mean, right now we basically deal with diagnostic scans of people who have already been diagnosed with dementia. And then we have to do a follow-up scan that is similar to or the same actually as their original scan. So we don't, mix apples and oranges, but actually starting them off on the right foot of the whole brain imaging would be quite amazing.
Yeah, no, I'd love that idea.
And knowing what we're dealing with, because, the problem is a lot of this stuff is, the full diagnostic is done upon death, which isn't very great. So being able to see what you're dealing with would make a huge difference. And could you use any of the imaging that we have, the pre and post imaging from other types of scans to actually build your model, or is that not possible?
Yeah, no, 100%. We can use historic scans. So we do that at clinics, and even, the data going back 18 years and things like that, we can do something with. Really quick, if there's any entrepreneurs in this space, what Liz is talking about, these preventative health or longevity, the shift in that direction is very interesting space to get traction in the marketplace because they are innovative physicians or clinics. So they are more willing to try something new. And that's a great opportunity, I think, to get into the marketplace because it's not easy to break in medicine, but at least these clinics are more open to that. So that's not something I learned about coming out of Stanford as a potential marketplace. It's something we had to figure out, but I would recommend that might be a nice go-to-market strategy or at least gateway.
Yeah, we definitely want to grow those industries with as many clinics as possible.
So for the way you interact with these clinics, they are doing whole body MRI. Do you just take that or do you need them to do a more specialized brain scan at higher res to apply your model?
That's a great question. So we do have them do an extra brain specific module. We try to keep the time to a minimum. So if they are doing full body, then we'll just do.
So they still
brain
run
on
the
top of
extra
that.
scan. You just applied the analysis model.
Exactly. That's it.
Okay.
Yeah.
So just a comment and maybe a question embedded in the comment. So besides the growing longevity clinic that are popping up, which are a sort of slightly different model, there's also this preventative medicine scanning model that's growing up at a lot of places, which are the same thing, but separately talked about. They're not called longevity clinics. This is Human Longevity Inc., is sort of a hybrid. And then there's QBio and Prenuvo and Peter Diamantis's one. And Peter Attia now is backing this one called Biograph. These are these clinics that do genetic analysis, full body MRI, and then a whole bunch of other, like the Biograph one does a whole bunch of cardio scans, like coronary calcium scans and stuff like that. This seems like another, maybe you're lumping that in with longevity clinics, but they're different from the kind of ones we see popping up that Andrea Mayer and others are talking about, and that the Wall Street Journal article you mentioned talked about. You should, if you're not already, you should probably be looking at partnering with those orgs too, because they could do the exact same things with their, just slightly more detailed brain scan and then apply your stuff. And that would just be an extra level of stuff they could give their customers, possibly for an extra.
I love that. Actually, most of those are already our clients without saying. Yeah. So you're exactly on. Yeah. So from a practical standpoint, we can work with these clinical images that Nicola was just mentioning, but we can also acquire very high-end type of images, and it depends on the clinic and the time you take. So that's all technical things, but it's very interesting.
To get the extra brain level detail you need, you have to wear that thing that holds your face in the, that like claustrophobic mask thing that people who do functional brain MRI type stuff do, or is it just a normal brain MRI experience for longer?
It's not so pleasant to get scanned. So that's why we try to minimize the time. The nice thing about that is the scanners have gotten so much faster and they continue to get faster and faster. So before it used to take, an hour when we were scanning patients back in my research days, and now it's down to 15 minutes or less. And where is this going to go in 5 years? So that's really exciting. But yeah, it's not pleasant. So let's get people in and out as quick as possible.
We have 3 more questions in the queue. Let's see how many we get. Let's see how fast we can move through them. First one, we have one from Hank. Do you want to unmute? Do you want me to read out?
Yeah. Hi. Great talk.
Hey.
I was just curious how direction you're trying to go with, or what is even capable of with the algorithm and training on like MRI detail. Are you ever going to be able to, try and predict, say, like dendritic spines or anything down to that level, even if you had a million gajillion Golgi stains trained on or something like that?
I mean, so that's a great question. So from a research perspective, and from the Lupus Center at Stanford, we're just going to push the scanner and keep patients in there forever. Maybe you can get a postmortem and do something with it and push further and further. But what I'm trying to emphasize here is the image quality we can acquire now can tell us so much about the patient's, the underlying systems impacting that patient and how that patient is aging. So going down deeper and deeper, it's going to happen, and that's going to be great, and we're going to push that. But the return you're going to get in terms of actually providing actionable information to that patient, and in terms of deciding what's the best treatment for an individual patient, isn't going to get that level of return anymore. So from a research side, let's do it. From a a company side and from a healthcare system side, there's less reason to do that. And you're seeing this a bit in the startup world. There's more and more companies developing low field scanners. So there are these small scanners that can acquire images quickly, and they're very cheap, and they can move the scanner around on a dolly. The point there is it's just even low quality images. There's still so much information there that's being underutilized, so you can actually use that in the clinic. But yeah, I do love going deeper and deeper. So I think that there's a space for that.
Cool. Thanks.
We're racing through here. Love it. Next one up, we have Paritosh.
Very good presentation, Owen. Thank you so much.
Thank you.
My question is mainly around the data validation steps and, the kind of data that you have trained the models on. So just differentiating structure of the brain, the topology and the connections. So what are the, each brain region has in-depth differentiation, as you already pointed out, based upon the patient and human phenotype. So, how deep you have gotten in each region, and what is the depth of visualization and the connection factors depending on differences in humans, like mutations, transformations of the data sets, to find similarities and outliers you have made depending on the silos of population and the demographics. Yeah, that's like kind of a broad with the question.
Yeah, no, I mean, that's a great question. Highly technical. I'm going to try to make it, if you want to dive deeper into the tech. BrainKey's perspective, we actually allow people to upload scans, and we'll create the 3D visualization for them. So we get scans from all over the world, and the quality of images, but also the brain abnormalities we might see are pretty large. So we've had to continuously adjust our algorithms to deal with that heterogeneity. So that makes it possible for the underlying data for us to do something with. A brain is a brain, but a patient might be missing a corpus callosum, or have a TBI, so their temporal lobe is just half missing on one side, or they had an epileptic problem and they removed part of the brain. So we do come across patients like this all the time, and so our algorithms have to adjust for that. The other interesting thing there is what defines a particular neuroanatomical region varies from neuroanatomist to neuroanatomist. There's close definitions there, but there can be slight variations. So that's another fun thing to talk about. But that's why, again, we like to draw back and look at this from these large systems perspectives and not look at, I think you can get so lost in the weeds when you just dive in closer and closer to very particular things rather than looking at the patient from the system's kind of outside perspective.
Yeah, just a quick follow-up. What are the validation steps you took to validate the data and the findings? Any conversations with, current leaders or?
I mean, we have multiple algorithms so that we could talk in detail about
Thank you.
each one, but we have publications around brain age or volumetric or things like that.
All right, let's see if we can get Aruna's question in as well.
Yeah, it's a quick question actually.
Sure.
So when you have patients older than 80, right? Do you
Yeah.
study healthy brains? Do you study if somebody is healthy for 90, 100? Why are they healthy? Like, why is their brain healthy? Do you study studies like that?
I mean, we love to. It's hard to get a bunch of people doing that.
Yeah, I
since,
can
there are-
imagine
yeah.
there'll be a lot of parameters, right? Health, their diet, their sleep, not having any trauma in life. So I was curious, like, how would you, say?
Yeah, so we'll get patients like that through our partners. We also have academic collaborations with, say, historical data sets where they do know what's happened with that patient. So hopefully they have some built in. And then there is a study we're not part of, but they are trying to collect patients that are 100 plus and looking at healthy patients that are, aging well. And we're really interested to see how that's going to turn out and if we can be part of that. But yeah, it's a great question.
Thank you. Okay, my final question in the final minute is the question that I always ask, which is, if people in this group are excited about your work, how can they help you? What can they do to support you? Are there any collaborations that you're currently looking for? Are you hiring? Are you fundraising? Is there anything in particular that would be useful to support you with?
So we're a startup, so we're never open if you're an investor, at least. The other thing, yeah, clinics, Liz, love to connect there. If anybody else has any ideas of potential partners for us, appreciate it. And if you're interested in neuro or AI in general, love to chat. One thing we didn't dive into too much from the We love incorporating other types of data. We actually incorporate genomic data in one of our newer reports, and we want to push that further and further. So genomics, epigenetics, things like that. Love to chat.
Awesome. Thank you so much. This was really insightful. I love that we had such a long Q&A. Thanks a lot for, basically like screen sharing, like flipping between different images. It was really fun. Thanks a lot for your talk. Very exciting stuff. Thanks everyone for your great questions, and I'll see you for the next one.